So next up is two presenters, Aaron Mills and Tandra Fraser, on things to consider when evaluating new crop inputs and biologicals. So Dr. Tandra Fraser is a soil microbiologist with Agriculture and Agri-Food Canada in Charlottetown. Her research focuses on soil health, nutrient cycling, and biological indicators of soil functions. Her experience prior to working for AAFC include research on phosphorus biochemistry and soil biodiversity at the University of Reading in the UK, with the Global Soil Biodiversity Initiative at Colorado State University. Tandra received her PhD from the University of Guelph and an MSc BSc, Bachelor of Science in Soil Science from the University of Saskatchewan. She grew up on a conventional grain farm in Saskatchewan. Is Aaron joining you?
Yeah, right.
Oh, there you are. Surprise, surprise. Dr. Aaron Mills has been a research scientist with AAFC Charlottetown since 2012. He holds degrees from NSAC, UPEI, and Dow. Most of his work is focused on different aspects of PEI cropping systems, including soil health measurements and overall system productivity. He is also working to generate local data to help growers to include new crops in the rotation, including emerging pulses, oilseeds, and grains. Thank you.
All right, great. Okay, thank you very much. First off, I just want to thank all the people that were helping us out with this. At the technical support, honestly, projects that we do like this are kind of daunting. They're huge, and we need really strong technical support to be able to pull this off. So these people were a massive help. And Heather and Ellen helped make this all happen. So I'm going to start off fresh after lunch with one of the more exciting things, the Fertilizer Act. Okay, so before 2013, believe it or not, companies had to prove efficacy of some of the fertilizer products that they were producing or planning to register. And actually, when I was doing my master's, I shared an office with one of the people who used to review the data, the multi-site data for these products. And really, it was a globally recognized system. So, you know, countries all over the world would basically say, well, if it's good in Canada, then it's probably okay for us. And but unfortunately, we moved away from that. And the Canadian government moved into safety-only testing. So these products, they no longer have to prove efficacy, but they have to show that they're free from pathogens, heavy metals, and environmental toxins. Okay. So why was merit testing eliminated? So it was part of regulatory modernization, more or less cutting red tape. And, you know, my big boss was here earlier talking about cutting red tape, so I don't really have to touch on that. But largely, initially, I guess what it was trying to do was to allow products to reach the market quicker. But what it actually ended up doing was shifting the burden of proof onto the manufacturer, but also the consumer. So it really is very much a buyer beware. So, you know, these products, they are registered, but they're registered to be free from pathogens, heavy metals, and environmental toxins. Okay? All right, so when we run these experiments, we really want to capture as much of the variability as possible. So we want to know, you know, are these products actually contributing to a treatment effect, or is it due to random factors? So the way that we do that is we account for the variability. And so with this arrow here, you can see the lowest amount of variability is in the rep. Oh yeah, wrong button. There we go. the rep. And so this is a replicated field experiment. And then site, that's like the field-to-field differences. And then finally year. So I don't really have to, tell farmers that every year is different, because every year can be drastically different. And it has the most, it kind of injects the most variability in a lot of the stuff that we're measuring. So basically, a single site in a single year, you can't draw any conclusions from that, because it's basically telling you nothing, right? So we try to overcome that through replication in our fields, but we still want to run the experiment for, at least 2 years, right? So that's kind of what we're shooting for. Okay, this. Okay, I've taken a lot of stats courses, and this one throws me into a deep rage every time I see this. This is essentially, and this is nobody's graph in particular. This was generated by ChatGPT. And you'll see a lot of this, and what these represent is a single site year. So these are 100 field sites. And essentially what this is, there's no statistics done on this whatsoever. It's just using a visual, like, you judge by how these means are and decide whether it's going to be a product that's good or not. And really, again, this is a fictitious slide, but, if it's 50% positive, well, that means that it's also 50% negative, and, the average of that is probably 0, right? So it's just, this is one thing that I just kind of want farmers to keep an eye out for, because really it's not analyzed data. It is data, but it's raw data, okay? All right, Tandra? Let's see, pointer at the top.
Thanks, Aaron. So I'm just going to take this opportunity to talk about one of the trials that we have running at Harrington Research Farm with the Grains Council. Biostimulants, it's kind of a catch-all term, so this is a lot of the products that are outside of fertilizer or pesticides, and they're typically biological products, and they can be applied to soil or plants or seeds, and they're supposed to improve plant growth. So a lot of the claims are that they stimulate natural processes, so this could be things like nutrient uptake, tolerance to abiotic stress, so something like drought, disease symptoms, and that this would all lead to improved crop quality and yield. So there's a whole bunch of different kinds. There's different humic and fulvic acids, there's proteins, seaweed and plant extracts, which is usually some sort of micronutrients or plant hormones, biopolymers such as chitin from crustaceans, or beneficial microbes. So this is mostly what I'm going to be talking about today. And one of the best examples of a commercial application is rhizobium inoculant, where for years producers have been able to apply this to their legume crops. They can reduce or eliminate their nitrogen use based on this and still maintain good yields. So these biostimulants, it's not a new idea. Just a quick search, and there was over 700 scientific papers from 2009 to 2019. But these aren't all field trials, right? This could include something that's been done in a petri dish to look at different functions. Could be greenhouse experiments, or it could be field experiments, and they're not always positive results. But microbes have evolved, right, to have these functions. If you look in nature, they're capable of doing these things. So the science behind these are there. It's just, it's sometimes difficult to get consistent yield benefits in the field. So the objective of this experiment was to look at a few different biostimulants on the market to see if we could maintain yields in wheat and corn under reduced nitrogen fertilizer. So can we take away some of the nitrogen that's being applied, use these products to either increase nitrogen uptake or fixation, and improve fertilizer use efficiency? And then we're also within this trying to reduce losses. So those are losses either to nitrous oxide to the atmosphere or to leaching. So if the nitrogen's leaving the system in this way, you're losing your money, right? So for the different treatments, we had a control, so no biostimulant. We had a seaweed product from Acadian Plant Health, so was mostly soluble potassium. We included Invita from Syngenta, so this is a bacteria that was isolated from sugarcane, and it came about because researchers noticed that it was producing more nitrogen than was in the soil, and that there was something else working there, so they were able to isolate this and then commercialize it. Nutrition nitrogen, which is a methylbacterium, it's also a nitrogen fixer, but a different bacteria. And then we tried to combine these two just to see if there was some sort of effect of the two bacteria, that if one was working and one wasn't, that if we could kind of equal that out. So just a little bit of the background on how these things are supposed to work. We have the nitrogen fixers, which are the Invita and Nutritia. We applied them as a foliar application, so they're meant to go on early in the morning. You want the plant at a fairly early stage, but enough so that there's some leaf tissue there for it to actually catch the product. But then that it will be early enough that it can kind of colonize through the plant and work. So this graphic here is just the nitrogen cycle, and it's from actually a legume crop. So this nitrogen fixation process is similar to that you see in the rhizobium in legumes, but it's supposed to be able to work on other crops outside of that. And it's not specific to colonizing the roots like you would see with rhizobium, where you get the nodulation on the new roots. So the idea of this is that you can provide the plant with nitrogen over the growing season, right? So typically if you're applying fertilizer at planting, it's a long time until you actually need that for the grain fill stage, which is when there's a lot of the nitrogen uptake there. So if you could combine these products that can fix their own nitrogen from the atmosphere, it's basically free nitrogen besides the cost of the product, and then you can maintain these yields. So in 2023, we set up a trial out at Harrington, and I'm a soil microbiologist, so I work with my colleague Dan McCackern, who's a biologist with us, and him and his team take care of all of the plot, the seeding and the maintenance and all of that. So we worked with 2 crops, corn and wheat, the 4 biostimulants plus the control, and then we also included 3 nitrogen rates. So we had 100% of the recommended rate for corn and wheat. This was applied as Super U, and then we tried to reduce that by 30% to see if we could maintain those same yields, so the 70% recommended, and then we had a 0 nitrogen control. So even though we don't expect a producer to grow corn with 0 nitrogen, we wanted to see if maybe we could stimulate the biostimulants to work under these extreme conditions, but also that we need this data to calculate the agronomic efficiency of these. And then we collected plant and soil samples throughout the growing season. So first off, the yield. So for silage corn, we only had the 2 years of data, and all of these graphs are set up in the same way, so that the yield is on the vertical y-axis, and then the biostimulants are along the bottom. So the control is always to the left, and then the dashed lines just show the comparison of the biostimulants to the control. So as you can see from these lines, at any of the nitrogen rates, so 0 is in blue, 70% is in orange, and then 100% in green, but we're not seeing any differences between the biostimulants and the control at any of the nitrogen rates. What is interesting here, and I think this was brought up earlier about nitrogen in the corn crops, but that between the 70% and the 100% rates, we're not seeing any boost there. So that kind of indicates that either nitrogen isn't a limiting factor here, or else we're optimizing out the plant growth. And it's likely that it's not the limiting factor, especially for corn. And it was just mentioned about it could be heat units, it could be moisture, it could be another nutrient that's actually limiting the yields in these crops. So we see the same trend when we look at the actual grain corn from 3 years. There's not a negative benefit of adding any of these biostimulants, but we just don't see the yield boost there. When we look at the wheat data, the story is exactly the same, and same with the 1,000 kernel weight. So we're seeing that nitrogen response between the 0 and the fertilized ones, not surprising at all. but no difference with the biostimulants here. So at this point, we're really questioning what we're doing. Are we applying the product right? Is it alive? You know, is there something that we're doing wrong in these trials that we're not seeing the responses? So we decided to take some, we use DNA a lot to look at bacterial and fungal communities in the soil. So we wanted to see if we could use these techniques to actually look at whether or not these products are out in the field after we apply them. So Dr. Ashok Samaraju, who works with me in my lab, he was able to, we can extract DNA from the plant samples, and then we can target the region specific to that strain so that it's only targeting that specific type of bacterium. And then we can amplify that so that we can actually look at whether or not the DNA is present in the field. So he started off with a simple growth chamber experiment where he grew some wheat. He applied it the same as we would in the field. And then over time, he did some sampling. And this was really interesting, actually, because so we have the specific primer sets for both Invita and the Utrisha. And he was able to see that, first of all, over time, so T1 there, that is before the application, so it wasn't present then. And then T2 was 7 days after, 14 days after, and then at the end point, so that was after grain filling, that he actually harvests those. And at harvest, he was able to separate the plant tissues and look at the leaf, the stem, the root, and the grain separately. And we see that it's all through the plant here. So it's not just on the plant from when we sprayed it on, but it's actually moved through there and down to the base. So we know that it's there in this situation and that it might decrease over time, but even at the harvest point in this controlled experiment, it was there. With Utrisha, we could also identify it there. Interestingly, it wasn't in the stem for some reason. So went to the leaf, to the root, whether or not it was just because it got sprayed on the soil and went down that way. But we can tell that it's there. And this is important because it's not like a rhizobium inoculant where you can dig up the plant root, you can look at the nodules, you can break them open, see if they're pink, and be fairly confident that the product's working. So after this, we also took the plant samples from the field, and we were able to show that they were also in the plots there. So we know that they're there. We're not seeing the yield boosts that we would like to, but it's possible that it's just that the improvements are just so small in the increments that we can't actually see that in the yield data. So when we look at the agronomic efficiency, so this is the yield of the fertilized plots minus the yield of the control plots over the fertilizer nitrogen. So this is kilograms of yield per kilogram of nitrogen. We see that with the 70% nitrogen, it's more agronomically efficient. I think we know this, especially with a lot of our commercial crops are not bred to really be efficient at taking up nutrients. As we apply more at 100%, we see that efficiency drop. And it's the same thing in wheat where we see with the control at 70% that we have the highest agronomic efficiency. So Dan did up some quick calculations of the net returns, and just like Steve said, this isn't profits. It doesn't include depreciation and overheads and all of that. But it's a comparison here that's important, where we have 70% nitrogen with the control. That's going to be your highest net return there. And for corn at least, then it's 100%, and when we have that control, that 0% nitrogen, your return isn't that high, not really surprising. And we did it for wheat as well, and again, it was same story, 70% nitrogen was your high, the biggest bang for your buck. But then what's interesting here is that we didn't see a huge difference between the 0 nitrogen and the 100% nitrogen as far as the net returns went. And you'll notice that the Invita and the Utrisha combined one, that one has the lowest net returns. And that's just because we were applying it at the full rate. So it just caused the treatment to be more expensive there. So I'm going to turn it back over to Aaron, but I'm happy to take any questions or comments at the end.
Okay, so So I tested some different products. This was probably 3 years ago we got this started, and it's the design's called a checkerboard, so we can evaluate a bunch of different combinations. And really where this came out of, we noticed there was a lot of growers in our yen who were using some of these emerging fertility products, and maybe there was probably 5 or 10% of them who were using the products and where they were supposed to be used, and everybody else was just kind of essentially just throwing it in the tank and going with it. So really what we wanted to do was test it in a controlled system to see if we were seeing a response. So we measured it in both spring wheat and soybeans, and so I'll show you an overhead picture of this design in a bit. It's a pretty gnarly design, but our main factor for the wheat, we wanted to look at nitrogen form because I know there's been a lot of questions out there about EENF, or enhanced efficiency nitrogen fertilizer, versus urea versus UAN, and so we wanted to include that as a main factor. And then with the soybean, because everybody knows that we don't put nitrogen on soybeans, we were looking at the inoculum. So we just used Celtec, which is just your regular rhizobium that we had laying around the farm, and we tested the Active Thrive, which is rhizobium and mycorrhizae, and then we also included this Optimize LV, which is rhizobium and lipoidooligosaccharide, whatever that is. Okay. And so with the wheat, this is our basic management approach. Everything got the same thing for all 3 years. We went with the old standard AC Walton. There's our seeding rate. And we put 70 units of nitrogen down pre-plant. And so it was either urea or Super U. And we top dressed with urea or UAN. So everything basically got the same amount of nitrogen, just different forms. And then just your regular management for most of it. With the soybeans, we put down a bit of K-Mag to help the magnesium and the sulfur a little bit. And we put these products in so I asked these companies for the recipe, and we were looking for something in as a tank mix with the herbicide, and something that's a tank mix with the fungicide. Okay, so these are the products that we tested, and it's not as balanced as I would have liked. We do have some deficiencies here that I'm going to talk about in a little bit, but this is the design overhead. This is the plot design. So basically, the way we have this set up is, we're applying mixtures going this way with the herbicides. I forget which one it is. Christian, do you remember? You're the one that did it? Yeah. And then we'd have the fungicides going the other way. Okay, so basically, all these plots had every combination, so we weren't just seeing the effects of, usually with some of the marketing stuff, you'll only see the company's results, but we wanted to know, well, is there any chance that we could get benefits from measuring multiple companies' products? And this is what it looked like from the drone. And if anybody's going to take pictures of this, take a picture of this one first, because this is where I talk about what's wrong with the study. So the cost, you notice before I had with the, we have two categories. So I have a herbicide category and a fungicide category. We also have some liquid that went down with the seed for some of those treatments and not others. And we also have fungicide treatments for some of them, and we don't have fungicide treatments for other ones, right? So we had to group together the herbicide treatments with the liquid in order to analyze it, and we didn't tease that out. So what you're going to see from the herbicide treatments is that the mixes are slightly overestimated because there's more products in there. And with the fungicide treatments, when we do the economics on it, it's going to be slightly underestimated because there's not one for every company. And then also, again, I did scientist math similar to Tandra, where I basically took the yield, multiplied it by the price that whatever was going around at the hospitality room at Christmas time, and minus the cost per acre. So the cost per acre is only the products that I used, not the fertility, nothing like that, okay? So that's basically as simple as it gets. Okay, so these are the deficiencies that I was talking about. So these products here, they would have been analyzed even though they were applied in furrow. They were counted or analyzed as part of the herbicide treatment, so it's going to increase the cost a little bit. And then here's this company's products. There's no fungicide treatment, so those are going to be slightly lower cost, right? Because there wasn't a treatment to go in with the fungicide. It's basically the same as the control. And I just wanted to show this slide to illustrate some of the year-to-year variability. We don't just take an average of all these. We actually account for the variability in the replicates and the years. Okay, so we'll talk about the wheat first. I don't think this is a surprise to anyone. Really, there's no difference in yield response for the source of nitrogen. Really, it's whatever form works best with your operation, and we're seeing that in the end too, where people are using a combination of urea and UAN, and even Super U is being used quite a bit too. Okay, so when we look at these products only as a herbicide, like as a tank mix with a herbicide, or only as a tank mix with a fungicide, this is what we're seeing for 3 years of data, and they were significantly different. But keep in mind, for example, this Alpine product, so this includes an in-furrow here, where the other companies didn't have that. And so when we put this bump chart together, we have, can I point? No, I got to point here, okay. So this is yield. These are ranked by yield, okay? So the good news is everything is better than the control, and we're ranging at a high of 1.65 to a low of 1.48 tons to the acre. But then when we do our scientist math on the margins here, these are also ranked. And so these ones here are better than the control, and these ones here are worse than the control. But there's not really a huge, we're going between, $500 versus $450 there, okay? Does everybody understand that? Yeah? Okay. Okay, so with the soybeans, same thing. There was no statistical difference between any of the inoculum treatments. And if you were to just add these products as a tank mix to the herbicide, or as a in-furrow application, or just to the fungicide, we did see a difference in yield compared to the control. And then here we have, again, it was good that the control was at the bottom, and this is how it's sorted out. Okay, so when I was going through, so this was 3 years ago, I got these recipes from these companies. No doubt the recipes have changed. Some of these products actually aren't even available when I was trying to find pricing for at least one of them. And really, we run into this all the time on the East Coast where we're adopting things from Western Canada that might be tested in other places, but really we need to be running more tests in under our environmental conditions. So we actually have 2 years of this left. And honestly, I would like to invite these companies to revisit this, and we can try and come up with, what are the best combinations that we can possibly put in for the next 2 years. We're doing a winter wheat checkerboard where we're measuring slightly different things, but we're definitely interested in finding out what the winner is from all the, like we could have inputs in on this if we want to. So in terms of applying this to your farm, I've got 53 seconds left. So first of all, we really want to kind of go back to what we're doing on the on-farm agronomy with the Atlantic Grains Council. We really need to keep it simple. One field in 1 year doesn't tell you anything. We're really, in terms of treatments, you want to look at essentially a split field, and you want to harvest as close to the line as you possibly can because that captures the most variability in the field. 1 year won't do it. 2 fields, 1 year is better than one, but you really want to get up around 10 or 15 before you can actually find a trend. And finally, if you need help with any of this or with design or implementation or anything like that, contact us, contact the Atlantic Grains Council, and we can help you out. Okay? That's it.